Okay. So this is the second video in the protein synthesis series. This will be the Wednesday videos. You already have one of them, so you should have watched transcription. This is translation, and then there'll be another set of videos, your like Friday videos, but the third set of videos that you're gonna watch. So translation involves taking your mRNA and turning it into a protein. You just watched a video on how we make mRNA, which was the transcription, taking it from DNA to an RNA. The things that we need are ribosomes, which is going to physically do the reading of the codon, tRNAs with the correct amino acid sequence, and mature mRNA. So ribosome structures. Ribosomes are made from proteins and rRNA, also known as ribosomal RNA. It has two subunits, a small subunit and a large subunit. And if we look, we'll see the small subunit sits on the bottom, the large sits on the top. And this is where the tRNA attaches to deposit whatever amino acid. If we look, we have the A site, the P site, and the E site. And basically, the A site is where the tRNA arrives with the correct amino acid. The P site has the tRNA that attaches the tRNA at the A site, and the E site is where the tRNA exits. So basically, I'll have my set of 3 codons that will sit in my A site. It will be read, and tRNA will arrive. It shifts the 3 codons to the P site where it's physically bonded, and then it shifts the 3 codons to the E site where the tRNA leaves, and the amino acid is now like bounded to the rest of the chain. If we look at the structure of the tRNA, so we've got my ribosome, we've got my string of mRNA, and then we have our tRNA coming in to read it. It looks like the image on the left. I don't know if there's any way I can really describe it, but basically what it uses is an anticodon to read the codon. So in that first A site, I've got my 3 letters, and the tRNA is going to arrive and use its anticodon to read it, and it's going to bring in the correct amino acid and attach it at the very top of this like key kind of structure. If we look, tRNA has like sets of 3 letters on the anticodon, similar to how all of these codons code for certain amino acids. So if I have GAC, it would be a CTC anticodon, which is going to tell me the amino acid that I need. In the E site, or sorry, the P site, so A is reading, P is attaching, the tRNA must get its amino acid by combining with a charging enzyme. This enzyme will put the correct amino acid onto the tRNA according to the anticodon. ATP is also needed. This happens in the P site. So from start to finish, the initiation of translation, there are technically 3 parts of translation: initiation, elongation, and termination. Initiation is the small subunit attaches to the mRNA, which is that base one we can see, at the AUG or the start codon. So it searches for that 3-letter sequence, and when it finds it, it attaches to that. Now the tRNA with the anticodon UAC attaches to the P site. So the tRNA is actually the next thing that attaches, and then the large subunit comes and sits on top. Initiation. Elongation. So the second tRNA arrives in the A site, attaches and reads it, and then brings in the correct amino acid. A peptide bond is formed between the amino acid and water is removed. So that happens at a P site. So we go from the A when tRNA arrives, move to P, use the bonding to create more of the amino chain, and then move to E, and the tRNA that's not being used anymore is removed while the amino acid chain stays. The final step is termination. At the stop codon, a stop protein will be produced, which is a releasing factor. and it arrives at the A site, so no more tRNAs can start filling in the A site. This means that once it gets to the final E space, it will all fall apart, and the polypeptide chain or the protein chain will go to wherever it's going. When we talk about going to wherever it's going, exported proteins, so proteins that have just been made, are tagged and they interact with signal recognition particles. It is responsible for synthesizing proteins into the lumen of the endoplasmic reticulum. So basically packaging and sending these proteins to where it needs to go. When we think about mutations during translation, mutations can definitely happen, and there are 3 types of base pair, which is like the sequence of letters. 3 types, and I think we've talked about this. So there's some that won't have any effect. You still make the same amino acid. So if we look all the way back to this chart, we see that, let's say like CUU, there's many different small mutations that could happen. You're still making the same amino acid, so nothing bad happens. We can have a missense mutation, which is a change in one amino acid, and so it plugs in a different amino acid, and then a nonsense mutation, which will code for a stop codon, and it'll shorten the chain altogether. So missense, you still get the same length, just one amino acid is incorrect. Nonsense, you're going to stop it early, and it's going to not be the correct protein. It's not going to continue making the protein. Frame shift mutations. So a base pair is a single letter. Frame shift mutations are mutations that result from the insertion or the deletion of a nucleotide. So the whole frame shifts. So if we think about it this way, if I have a sequence of 3, and instead of switching something, I totally take one of those amino acids out, the first amino acid of the next codon is going to be now read as the last amino acid or nucleotide of that codon. And so it totally shifts how we read it, therefore making everything after the fact potentially different. The missense, a change in one amino acid sequence impacts the rest of the chain. So if we look at the top one, the U is missing, and so now I'm reading all of my codons differently. Insertions or deletions of the full 3 letters will actually correct this issue because we are maintaining the frame, right? So the codons, the sets of 3. Again, so missense would change the rest of the sequence. Nonsense, you add a letter or delete a letter and you get a stop codon and the whole thing stops. That is the end of this and the end of transcription and translation. The next content we talk about will be totally different.